Great, thank you so much for having me here. I'm going to be talking a little bit about ecology and situations where ecology and evolution are coupled and act on sort of comparable timescales. Okay, so a little bit of a motivation. So, bacteria and phage are sort of, some of the biggest or most abundant sort of creatures or organisms or whatever you want to call phage on our planet. With enormous population sizes in the oceans, millions of bacteria and phage per milliliter of seawater. And in addition, these bacteria and phage are sort of very strongly interacting with each other. Some large fraction of bacteria are turned over every day by phage killing in the ocean. And yet, despite this sort of strong interactions, they sort of exist at relatively stable sort of population sizes. So I'm going to ask the question, what is sort of like the simplest way we can model these dynamics? Okay, so I'm going to propose a sort of simple model of bacteria phage ecology in which we sort of have a well-mixed situation, so maybe a milliliter of phage and bacteria. And these phage and bacteria have different genotypes that sort of enable them to interact with each other in sort of different ways. And the bacteria can grow of their own volition or by eating nutrients and processing metabolites. And without consuming bacteria, the phage will die at some rate. And we can imagine there are many different types of strains that are interacting with each other. And so then we need to sort of worry about these interactions, which if you have 2 to the L sort of bacteria and 2 to the L different phage, that's many different interactions. And so that's hard to sort of capture sort of at the individual interaction level. But what we can do is we can sort of assume that these interactions are drawn from some sort of distribution with some sort of mean and variance. And so, just as for future slides, a couple of important parameters that I'm going to sort of define here is A, which sort of measures the mean productive infection rate of phage, and sigma is sort of the sort of variance of this distribution of interaction rates that is sort of like a proxy for the potential diversity, phenotypic diversity, or the sort of like complexity of this ecosystem. And so if we sort of, model these interactions in a sort of stochastic interaction framework, the sort of simple model ends up failing spectacularly. So before I had shown these sort of experiments where, we sort of have bacteria and phage growing in a flask, and they sort of seem to be pretty stable in their population size over time. But if you sort of try to model this and incorporate diversity, which is natural in these sort of bacteria phage populations, the simulations just fail. So either the bacteria or the phage or both go extinct on very rapid timescales when you have a sort of diverse ecosystem that you're trying to simulate. So then a natural question to ask is, what's missing from this sort of simple model? And, it could be a lot of things, but what I'm going to suggest is, one of the really important things that's missing is that bacteria and phage both evolve on really rapid timescales. And so, what I'm showing here is that, a similar experiment from actually the same group as the previous experiment, where a phage sort of over time kind of gains the ability to recognize a new receptor on E. coli. If you look at the time scale of this, the final ABCDE genotype is the one that is able to recognize this new receptor. It does so in 10 days, which is very quickly. And so what we can do is we can go back to the model, and we can incorporate evolutionary dynamics. And so, once again, I'm going to, because I'm a theorist, I tend to simplify things. But the sort of simplest model that we can sort of propose to kind of account for evolution is a sort of bit flip type of mutation where different sort of strains have these sort of binary genomes that can sort of mutate an individual position during a given sort of birth event. And so by doing this, we introduce a new parameter, which is which I denote P, which is the probability that a birth event results in a mutation. And so if we sort of simulate this, once again, run some stochastic simulations of this process, the population dynamics can be qualitatively different. So if we sort of have neutral interactions, so the situation in which these sort of interactions between all the different phage strains and bacterial strains are identical, we sort of have the same problem. The population sort of crashes out on a sort of on a relatively quick time scale. But if we have sort of sufficiently variable interactions, so sort of high variance interactions, the populations are actually stabilized. And so it seems like you kind of need diversity to have stability in this sort of model phage bacterial ecosystem. But then if we look under the hood at all the different strains that sort of make up this these kind of bacteriophage communities, the different sort of genotypes or strains are fluctuating wildly, even though the population as a whole is sort of stable. And so, I've shown you one sort of simulation or one example of a simulation here. And you might say, maybe this is just a fluke. You just chose a certain parameter set that is that just happens to be stable. And so what we can do is we can actually vary this parameter P over many, many orders of magnitude. We can vary this parameter sigma over various orders of magnitude. So the mutation rate and the sort of diversity or the variability of the interactions. And then we can sort of plot both the sort of diversity of the community at the end of a simulation and the probability of extinction. And what we see is that there's actually a sort of sharp transition. So, for high diversity, so high sigma and high P, we both see that the final community is both very very diverse on the left, if we look at sort of the entropy of the different strains that are present. And moreover, that the probability of extinction is really low. So if we look on the right for very high sigma, the probability of extinction goes pretty much to zero. And so, a natural question then is, can we start to understand this transition both qualitatively and quantitatively? And I'm primarily going to talk about qualitative things today, but if you're interested in the more sort of mathematical analysis or the quantitative analysis, feel free to check out my preprint, which I'll link at the end. But so in pictures, I'll sort of describe the sort of mechanism of the sort of stabilization in this sort of predator-prey model system. So, without evolution, there's this sort of negative feedback loop that I've called a predator death spiral, whereby a predator will sort of gorge on some prey, which will enable it to increase its abundance, which will allow it to continue to gorge on this prey, eventually killing it and eventually driving it to extinction. But with evolution, the situation is quite different. So evolution sort of ends up ameliorating this death spiral. So the predator gorges on prey once again, but now instead of just increasing its abundance, it also mutates. So it creates mutants. And these mutants, they tend to have preferences that are quite different from their parents. And so, we've all been teenagers and we know we have very different preferences from our parents. And And this sort of difference in preferences sort of allows the prey to recover so that the death spiral is avoided. You know, we can, through some mathematical analysis, which I won't go over in this talk because it's so short, we can sort of derive the sort of phase boundaries between these sort of stable and extinct phases in terms of sort of measurable or potentially measurable parameters. So I define this composite parameter T, which defines the sort of boundary between stability and extinction. And if we go back to these sort of plots from before, we see that, you know, this parameter T, in fact, does sort of up to order 1 factors predict the sort of the boundary between the extinct and the stable phase. We can map out a phase diagram in which we notice the sort of the size of the sort of stable region kind of grows with the population size. So larger populations can be sort of expected to be more stable. And finally, in words, we can sort of interpret this sort of parameter T as a mutation rate times a population size, so that T can be sort of thought of as a number of mutants per generation, so that the sort of stable phase corresponds to the part of parameter space where you have multiple competing mutants in the population at once. And this is known as sort of clonal interference in the sort of population genetics literature. And so what we can do, I'm almost done, is we can sort of compare this model to sort of population genetic models of rapid adaptation, which sort of predict a sort of constant population size or a constant fitness distribution that sort of travels according to a traveling wave in genotype space. Indeed, we see that this is not the case in our model. So if we plot the mean and the variance of the fitness distribution over time, we see that it fluctuates greatly. And, you know, to summarize, you know, evolution can stabilize complex communities. This multiple mutations regime seems to be the sort of regime of stability. And finally, you know, this model kind of departs from sort of classical models of rapid adaptation in sort of quantitative ways. So thank you.